Audio Jungle.
Cardiac cycle. Cardiac cycle is the complete sequence of events in one heartbeat, 0.8 seconds. Number 2. Systole is the period of contraction of the heart muscle. Number 3. Diastole is the period of relaxation of the heart muscles. Number 4. There are three main stages. A. Atrial systole for 0.1 second. B. Ventricular systole for 0.3 second. C. Atrial ventricular diastole 0.4 second. Number 5. Before atrial systole, during the late atrial ventricular diastole, the heart four chambers are filled with 70% blood. During atrial systole, A. Simultaneous contraction of both atria upon excitation of sinoatrial nodes. B. Volume in atria decreases while pressure increases. C. Atrial ventricular valve opens and blood is forced into the ventricles. Ventricular systole. A. Simultaneous contraction of both ventricles upon excitation of atrial ventricular node. B. Impulse transmitted along bundle of His and Purkinje fibers causes rapid contraction of both ventricles from the apex up. C. Volume in the ventricles decreases while pressure increases. D. Blood pushes against the AV valve and the first heart sound, loud, is produced. During atrial ventricular diastole, A. Both atria and ventricles are relaxed. B. Volume in both atria and ventricles increases while pressure decreases. C. Higher pressure in pulmonary artery and aorta than the ventricles pushes against the semilunar valves and second heart sound duct is produced. D. Blood keep on filling the ventricles until about 70% of the heart total volume. Number 9. Then cardiac cycles begins again. Regulation of heartbeat. Number 1. The heartbeat can be regulated intrinsically or extrinsically. Number 2. In intrinsic control, A. Impulse is sent to sinoatrial node or pacemaker and causes both atria to contract simultaneously. It delays for 0.1 second. B. Impulse is then transmitted to atrioventricular node, a long bundle of His and Purkinje fibers, and causes both ventricles to contract simultaneously from the apex up. Number 3. In extrinsic control, A. Heartbeat rate is controlled by autonomic nervous system, sympathetic or parasympathetic. B. Rate is increased by cardiac accelerator center in the medulla upon receiving impulse from the stretch receptor at the vena cava through sympathetic nerve to stimulate sinoatrial node. C. Rate is also increased by hormonal regulation of noradrenaline, adrenaline, and thyroxine to stimulate sinoatrial node. D. Rate is decreased by cardiac inhibitory center in the medulla upon receiving impulse from the stretch receptor at the aortic and carotid bodies through parasympathetic nerve to sinoatrial node. pressure is due to the accumulation of mineral salts in the root xylem. Mineral salts are actively loaded into the root xylem by the endodermal cells. In apoplast pathway, look carefully how water diffuses by osmosis down the water potential gradient from one root cell to another via the cell wall and extracellular spaces. But this pathway, it is blocked at the endodermal cells. The Casparian strip contains suberin, which is impervious to water. In symplast pathway, look carefully how water diffuses by osmosis down the water potential gradient from one cytoplasm of root cell to another via the plasmodesmata. In vacuolar pathway or transmembrane pathway, look carefully how water diffuses by osmosis down the water potential gradient from one vacuole of the root cell to another via the plasma membrane and plasmodesmata. Cohesion tension theory consists of three major forces. Number 1, cohesion forces of water molecules. Number 2, adhesive forces of water molecules to the wall of stem xylem. And number 3, transpiration pull as the pulling factor of water from the leaves. Cohesion tension theory describes the transportation of water in the stem xylem in a continuous column of water that gives rise to capillary effect. Remember, continuous column of water. Transpiration pool as described by Campbell 12th edition. Water vapor diffuses from the moist air spaces of the leaf to the drier air outside via the stomata. At first, the water vapor lost by transpiration is replaced by evaporation from the water film that coats the mesophyll cells. The evaporation of water film causes the air-water interface to retreat further into the cell wall and to become more curved. This curvature increases the surface tension and the rate of transpiration. The increased surface tension pulls water from the surrounding cells and air spaces. How they pull from surrounding cells are through apoplast, symplast, and vacuolar pathway. Water then from the xylem is pulled into the surrounding cells and air spaces to replace the water that was lost. At their source, phloem loading occurs. Sucrose is actively loaded from the source cell to the companion cell and into the sieve tube element. This increases the solute potential in the sieve tube and lowers the water potential. Water then diffuses in by osmosis into the sieve tube from the neighboring xylem, thus creating a high hydrostatic pressure in the sieve tube of the source. At the sink, phloem unloading occurs. Sucrose is actively unloaded from the sieve tube element to the companion cell and finally into the sink cell. This lowers the solute potential in the sieve tube and increases the water potential. Water diffuses out by osmosis from the sieve tube into the neighboring xylem, thus creating a low hydrostatic pressure in the sieve tube of the sink. Due to the difference in hydrostatic pressure gradient of the source and sink, the phloem sac is translocated from the source sieve tube to the sink sieve tube in a bulk or mass. It is unidirectional because it is from the source to the sink. Point to take note: if the sink is a sugar beet or sugar cane, then sucrose is transported against the concentration gradient by active transport. If the sink is potato tuber, then sucrose is transported by facilitated diffusion down the concentration gradient as sucrose is converted to starch to be stored. And finally, if the sink is germinating part or growing parts, then sucrose is transported by facilitated diffusion down the concentration gradient as sucrose is utilized for plants' metabolism. Potassium ion are actively transported into the sieve tube element from the companion cell. The potassium ion tend to accumulate near the sieve plate, increases the positive charges there, forming an electropotential gradient at the sieve plate. Due to the difference in the potential gradient, the potassium ions speed across the sieve plate, bringing along the water molecules and dissolve solutes into the next sieve tube. Potassium ion is then transported back into the companion cell. Water and dissolved solids circulate in a circular motion in the sieve tube element. At the sieve plate, the motion slows down and the kinetic energy acquired through motion forces water and dissolved solids to move through the sieve plate into the next sieve tube element. It is bidirectional. Fine cytoplasmic filaments, which contain phloem sac, are continuous in the sieve tube elements. Constriction and relaxation of the filaments pushing the phloem sac from one sieve tube element to the next. The peristaltic movement that uses metabolic energy form patterns of wave known as peristaltic wave.